Now let's take a look at those critical components that help regulate biochemical reaction, and that involves enzymes. Enzymes are biological catalysts. Recall that a catalyst is anything that speeds up the rate of a chemical reaction. So if it's a biological molecule, then it is a biological catalyst. When we have a chemical reaction that's driven by an enzyme, the reactants are now called substrates. So instead of reactants going to products, we have substrates going to products. The enzyme and the substrate bind to one another. The substrate is then modified and is released as products. This then allows this enzyme to be used again, and it could pick up another substrate and run another reaction. Most enzymes are proteins. Here we see a three-dimensional model of a protein. And there's an area on the protein that is called the active site. We see the active site up here where the substrate binds, and here in our three-dimensional we have this colored area representing the active site. Recall that proteins are made up of amino acids, and the three-dimensional shape of the protein is partly determined by the sequence of those amino acids in that chain. The amino acids of the active site are particularly important for the way that the enzyme functions, because these amino acids can provide the right environment. They might provide a proton depending on the type of amino acid. These amino acids will often interact with the substrates or create the right environment for the substrate for the reaction to occur. The cellular environment is also very important for enzyme function. If we don't have the correct temperature, it can cause the reaction to either occur too slowly, or if we have excessive temperatures, it can cause the enzyme to actually change shape, which is known as denaturing. And if the enzyme changes shape, it won't work correctly. The pH also can influence the binding of the enzyme and the substrate to one another and reduce the effectiveness of this enzyme. We typically, for enzymes, will have an optimal pH and an optimal temperature in which they work best. And if we are outside of that optimal range, they don't work as well. Enzyme and substrates are typically very specific. The binding site shape is very specific so that you don't have one enzyme running lots of different reactions. You typically have an enzyme that runs a particular reaction. The binding of the two forms what we call an enzyme-substrate complex, and different enzymes will catalyze different types of reactions. Here we see an enzyme. Here's its active site. The substrate comes in and binds to that active site, and then here it's releasing the products. In the middle here, where they are bound, we call that the enzyme-substrate complex. We see here two different types of reactions that are being catalyzed by the enzymes. We see one that is a building reaction, which is on the bottom here, anabolism. So we're taking two substrates, we're hooking them together to create a larger molecule product. So that is anabolism, where we are binding together substrates to form larger products. And then we also can break them down, which is shown on the top. So we take this molecule here, we break it apart, and we create smaller products from that. When we talk about the way that an enzyme substrate fits together, we often talk about the induced fit model of enzyme function. What that means is that when the enzyme and the substrate contact one another, it causes a slight change in the shape of the enzyme. So notice here the substrate is going to come in and it's going to bind. But when it does, notice right here, this enzyme substrate complex has caused a slight change in the shape of the enzyme. So this doesn't look exactly like it did over here. And that's that induced fit idea. These slight changes at the active site probably help to maximize that catalysis process. Induced fit is a relatively recent discovery. Prior to that, they had the lock and key model. So the lock and key model would have been very similar to this, but it wouldn't have predicted this change in shape right here. We still know that they are very specific, sort of like a lock and key, but once the lock and key fit in there, then that enzyme does change shape just a bit in the induced fit model. This shows us a three-dimensional image of an induced fit. So we have the two substrates coming in. As you can see here, the shape isn't perfect, but then in induced fit, it wraps around them and causes them to fit together very well. And so we're seeing over here these two molecules coming into this three-dimensional structure, and then we see the protein sort of folding around it a little bit. And that helps that catalysis that occurs in that active site. Enzymes increase the rate of chemical reactions by supplying or lowering the activation energy required for the reaction. Remember that activation energy was that hill that we have to overcome in order to let the reaction go. How is it that enzymes do this? How do they allow the substrate to reach this transition state so that it lowers that activation energy and allows the reaction to occur? And it can happen in several different ways. One is it might take those two substrates and position them in just the right way so that they align, allowing for the formation, let's say, of this bond. So if we take the two parts of the molecules that are supposed to bind to one another and we put them together just right, they're more likely to form that bond rather than relying on them randomly positioning themselves if they just bump into each other to align correctly. They also might provide the right environment. So it could be that a certain chemical reaction requires an acidic environment or a slightly polar environment. And so inside that active site, you can think of it as being an area where we might create just the right environment for that reaction to occur. It also could make it so that it takes the substrate and stresses it a little bit so that it's a little bit less stable. Let's say imagine we're breaking apart a molecule. And so we bring in this disaccharide, let's say, and then it binds to the enzyme, and then that enzyme does this induced fit where it flexes a little bit. And when it does, maybe it puts pressure on that bond between those two sugars, allowing it to break more easily. It also can temporarily react with the substrate and actually chemically change the substrate. So it might not just be a physical interaction. There might be a chemical interaction where it maybe gives it a proton or takes a proton away and allows it to react that way. After catalysis, remember, the product is released and then that enzyme is available to catalyze another reaction. So these enzymes can be used over and over again, meaning that enzymes are not required in very large quantities typically. This video will give us an overview of enzyme function.
Every single second, inside every living cell, thousands of chemical reactions are taking place. These reactions are performed by enzymes. An enzyme is a protein that catalyzes a chemical reaction. It initiates the reaction, speeds up the reaction's progress, and makes sure the outcome is always the same. These enzymes often work together to form longer pathways, such as the citric acid cycle, which is a series of chemical reactions used by cells to generate energy from carbohydrates. The essential tasks of life, such as metabolism, protein synthesis, and cell renewal and growth, are all regulated by enzymes. The life-sustaining power of enzymes lies in the fact that they catalyze reactions in mild conditions of pH, temperature, and atmospheric pressure. The rates of catalyzed reactions are millions to trillions times faster than those of the same reactions uncatalyzed. To speed up a reaction in the absence of enzyme, additional energy would need to be provided as heat, which jostles the substrates and occasionally provides enough energy to trigger a reaction. In the course of most reactions, an unstable and highly energetic transition state is formed as the substrates are transformed into products. An enzyme acts as a template for the reaction, binding to its substrate and holding it in the proper position to form the product. An enzyme also surrounds its substrate with reactive groups that stabilize the transition state, making it easier for the reaction to occur. To understand how enzymes work, let's take a closer look at a reaction in the citric acid cycle that is catalyzed by the enzyme aconitase. Aconitase binds to its substrate citrate and removes a hydroxyl group and a hydrogen atom to form the intermediate cis-aconitate. It then adds the hydrogen and hydroxyl back in slightly different positions to form the product isocitrate. In the active site, some amino acids are perfectly positioned to recognize the substrate and hold it in the optimal position for the reaction to begin. Some amino acids are involved in recognizing and holding the substrate. Other amino acids are directly involved in catalysis. Histidine 101 acts as an acid by donating its proton. Thanks to the chemical environment of serine 642, it can act as a base by accepting the proton from the substrate. The active site of aconitase also contains an iron-sulfur cluster that stabilizes the substrate electrostatically and helps to position it relative to the catalytic residues. The first step in the reaction is dehydration. In this step, histidine acts as an acid and protonates the hydroxyl on the substrate, allowing it to leave as a water molecule. Serine then acts as a base, extracting a hydrogen atom from the opposite side of the substrate, forming the intermediate cis-aconitate. Cis-aconitate then flips upside down, and the complementary hydration reaction is performed. In this step, histidine grabs a hydrogen atom from a passing water molecule, placing the resulting hydroxyl group back onto the substrate. Serine then returns its hydrogen atom, and the final product isocitrate is released. Notice that the enzyme itself was not changed by the reaction. It extracted a hydroxyl group and a hydrogen atom, and then put them back, thus starting and ending in the same state. This is the hallmark of a catalyst. When it finishes a reaction, it is ready for the next, so it can perform thousands of reactions in a row. Notice also that the shape of an active site is often flexible. Many enzymes surround their substrates, enclosing them to form the perfect environment for a reaction. Enzymes are fundamental to life on Earth, working every second of every day to maintain life processes in every cell in every living creature on the planet.
Let's take a look at the factors that affect enzymes. We've said that enzymes require a particular environment to do well, certain pH, certain temperature. Regulation of enzyme activity helps cells control their environment to meet specific needs. For example, digestive cells in your stomach will work harder after a meal than they will when you're asleep. So we have to regulate when those enzymes are being produced. Enzymes can be regulated by a variety of factors. Many of these are modifications to temperature and pH, as we've mentioned. We also can have molecules that inhibit or promote enzyme function. There are also other molecules that can interact with enzymes that allow them to function properly. These can be either coenzymes or cofactors. Coenzymes tend to be large biological compounds that interact with enzymes, and cofactors tend to be metal ions that will interact with enzymes, allowing them to function correctly. Here we'll take a look at some factors that affect enzyme function.
Three important factors which impact the rate at which enzymes catalyze reactions are substrate concentration, temperature, and pH. Substrate concentration is important because at very low concentrations, many enzymes are just waiting around for a substrate. At high concentrations, all the enzymes are hard at work, and the extra substrates are waiting around for a free enzyme. So as substrate concentration increases, the amount of enzyme activity saturates, as shown in this graph. In addition, the rate of activity of an enzyme is highest within its optimal temperature range. Enzyme activity occurs randomly when substrate and enzyme bump together and bind. At low temperatures, molecules have low kinetic energy and are unlikely to randomly bump into each other. As temperature increases, the random interactions happen more frequently. As the temperature increases past a certain point, however, the enzymes start to denature, or fall apart, resulting in a decrease in enzyme activity. Enzymes also typically work best in a narrow pH range. pH affects enzyme activity by affecting the structure of the enzymes themselves. Enzymes have many ionizable side chains and prosthetic groups, which affect the intermolecular bonds which hold together their secondary and tertiary structure. Maintaining the correct secondary and tertiary structure is critical for the proper alignment and functioning of the active site. Outside of an optimal pH range, changes to ionizable side chains can break bonds holding together secondary and tertiary structures, causing the active site to fall apart, and decreasing enzymatic activity. In addition, the substrate binding to the enzyme might have its own ionizable side chains, which can also be affected by pH. Substrate concentration, temperature, and pH are not the only factors affecting enzyme activity. Cells also have some control over enzymatic activity. For example, they can assert control through the use of post-translational mechanisms such as phosphorylation and glycosylation, each of which can increase or decrease enzymatic activity. In addition, cells in different organs produce different enzymes, and each cell has control over how much of each enzyme it produces, as well as which enzymes go to which organelles. For example, digestive enzymes are found in the stomach, but not in the brain. There are also substances that increase enzymatic activity, called activators, and substances that decrease enzymatic activity, called inhibitors. Inhibitors can interact either reversibly or irreversibly with enzymes. Reversible inhibitors interact non-covalently with enzymes. Irreversible inhibitors usually associate with the enzyme through covalent interactions, and so decrease the concentration of active enzyme. There are three major categories of reversible inhibitors: competitive, non-competitive, and uncompetitive. Normally, a substrate binds to an enzyme at the active site, after which the enzyme catalyzes a reaction and releases the products. Competitive inhibitors compete with the substrate for access to the active site, where they bind the enzyme. Hence, they can only bind to free enzymes, which are not bound to any substrate. Non-competitive inhibitors not only bind to free enzymes, but also to those bound to substrates. This means that they bind not to the active site, but to the so-called allosteric site. In doing so, they cause a conformational change that increases or decreases activity of the enzyme. Uncompetitive inhibitors only bind to enzymes when the enzymes are bound to substrates, and they bind in such a way that the enzyme cannot release its products.
Now let's take a look at enzyme regulation. In enzyme regulation, we can have another substance that will come and perhaps turn the enzyme on, turn the enzyme off, or maybe just regulate the rate at which the enzyme works, speeding it up or slowing it down. Here we're looking at two different types of regulation. Both of them are allosteric. What allosteric means is that this substance will bind to the enzyme at a place on the enzyme that is not the active site. It's a different place on the enzyme. And when it binds, it might inhibit that enzyme or it might activate that enzyme. In an allosteric inhibitor that we see here on the left, this molecule binds. Prior to binding, that enzyme active site was the right shape for the substrate. So the substrate could have come in here and bound to this enzyme. But when the inhibitor binds to the enzyme, it changes the shape of that active site. Now the substrate can't bind. So we are inhibiting that function of that enzyme. On the right, we see the allosteric activator. When the activator is not bound to the enzyme, the molecule cannot bind to its substrate. When we add the activator, it now changes the shape of the active site so it can bind to the substrate and it's ready to catalyze that reaction. So we either turn it off or we can turn it on. Here we'll see a short video on enzyme regulation.
Our cells are amazing molecular machines. Every day, they convert food into energy, such as ATP, that drives the millions of biochemical processes necessary for keeping us alive. The pathways used to convert substrates, such as glucose, into products are collectively referred to as metabolic pathways. The drivers of these metabolic pathways are enzymes. Enzymes are usually proteins that work to assist chemical reactions by building or breaking down molecules. However, enzymes don't drive reactions at a constant rate. The rate of reaction can speed up, slow down, or stop completely according to the cell's needs. This process is analogous to an assembly line where raw materials are converted into products. The assembly line must be monitored to ensure that the supply of products does not exceed demand. If products are being created at a rate that is faster than they can be used, both resources and products are being wasted. The assembly line can produce at a slower rate or stop completely to prevent waste. In cells, this process is called feedback inhibition. Feedback inhibition is achieved through a process called allosteric regulation and plays an important role in many metabolic pathways. Enzymes have a unique area called the active site where substrates can bind. Enzymes that can be regulated have an additional area called the allosteric site. Downstream products can bind to this regulatory site, causing the enzyme to change shape and prevent it from binding with its substrates. This causes the entire pathway to halt, and new products are no longer formed. Conversely, when the amount of downstream product gets too low, the enzymes interact with their usual substrates and begin forming products again. Feedback inhibition plays a critical role in preventing cells from wasting resources and energy. Mutated enzymes that do not respond to allosteric regulation have been linked to disease states such as cancer. Many processes in our bodies rely on feedback inhibition to keep everything running smoothly and efficiently, maintaining homeostasis.
Let's look a little bit more at enzyme inhibition. In addition to having an allosteric inhibitor, we can have other types of inhibitors. If it's a competitive inhibitor, it means that that inhibitor has a very similar shape to the substrate. So it competes with the substrate for the active site. If the inhibitor comes in and binds to the active site, then the substrate can't bind to it. Now, a non-competitive inhibitor will bind to a location, perhaps not right on the active site, but it might be somewhat blocking the active site, or at least making it so that the enzyme doesn't have as easy access to the active site and slows down the reaction. So in competitive, the two compete for the same site. In non-competitive, the inhibitor comes in and binds to an area maybe near the active site, but not on the active site, but then slows down the reaction rate. Competitive inhibitors slow the reaction rate down, but it doesn't really affect the overall maximum rate. Here we see a reaction in purple where you have a normal enzyme that's not being inhibited and the reaction rate. When you have a competitive inhibitor, we see that the reaction rate is slower, will reach the maximum rate. It just does it at a higher substrate concentration. Remember, it's competing. How could we increase the rate of a reaction if it's competing with the inhibitor for the active site? If I flood that with substrate, if I give it tons of substrate and I only have a few inhibitors, then it's more likely that when the molecule gets there, the one that arrives at the active site will be the substrate and not the inhibitor. If I give it a whole bunch of substrate, then I'm going to get the reaction to occur pretty quickly. The way to increase my reaction rate if I have a competitive inhibitor is to add a whole bunch more substrate. That doesn't work in the non-competitive state because the non-competitive one isn't binding to the same place. It's binding somewhere else and then causing the substrate to not be able to bind to. If I have a non-competitive inhibitor, even if I increase this substrate concentration, I'm not going to get as fast a reaction, no matter how much substrate I give it. So this is something that you can do in a lab to determine if a inhibitor is competitive or non-competitive by increasing the substrate concentration and seeing if that has any effect on your overall rate of reaction. And the maximal rate is the rate that we see here up at the very top where it levels off, where the substrate is not limited. With the normal enzyme, obviously the more substrate I give it, the faster I create products, up to a certain speed. That maximal rate is typically going to be determined by how quickly the enzyme can put them together and let them go. If I give it too much substrate and all the enzymes are currently working and there's no other enzymes available, then I'm going to hit some maximum rate because it can't go any faster. All my enzymes are currently working. I don't have any more workers, and that's where that maximal rate would be. So out here, if I increase the enzyme concentration, then I could get it to go up a little bit more because now I have more enzymes working to create that reaction. We mentioned a few minutes ago about enzyme cofactors. Some enzymes require one or maybe several cofactors or possibly coenzymes to work. Coenzymes are typically large organic compounds like these vitamins that you see here. The vitamins in our diet typically are functioning, many of them, as coenzymes for these enzymes to work correctly. So in the absence of the coenzyme, it's essentially not functional, and then you add the coenzyme to the enzyme and it becomes functional. And that's also one reason why we don't typically need large quantities of these in our diet, because we don't, again, need large quantities of enzymes. We use them over and over again. However, if we have a deficiency here, we can see that a lot of chemical reactions might not run, and that would be certainly a problem. We also have cofactors. Cofactors tend to be inorganic ions like iron, magnesium, zinc. They tend to also associate with some enzymes, making them function properly. For instance, DNA polymerase requires zinc ions in order to work correctly. And DNA polymerase is an enzyme that helps us build DNA molecules. Some other example of coenzymes, ATP can serve as a coenzyme, NADH, and then of course the vitamins that we see here. These molecules are primarily brought in through the diet. Now let's take a look at this concept of feedback inhibition. In a metabolic pathway, we want to have a way of regulating how much product we make. If we make too much product, then it can pile up and become either a problem in the cell because we have too much of it, or at least make it so that the cell is very inefficient because we're making things we don't need. What we do is we essentially look at the amount of substrates and products that we have, and as the amount of product starts to build up in the cell, we want a way of turning that machinery off temporarily until we use those products and need more of them. And that's what feedback inhibition is all about. What we have here is we have a metabolic pathway, so several different enzymes, different reactants and products for each of these different steps. In our initial one, the substrate comes in, it binds, and we go through lots of different processes, but now here at the end, we have our end product. And this end product comes back around and binds to an enzyme, typically an enzyme that is very early here in this metabolic pathway, and essentially turns it off. So when we have a buildup of end product, we turn off one of these enzymes that happens early in the pathway and essentially shut down this machinery temporarily. Once we start using up this product, then obviously the concentration of this product will start to go down in the cell, and this will then make it so that the enzymes work again because they won't be bound to these enzymes. This all works because metabolic pathways are these series of reactions, and we have different enzymes catalyzing each step of the reaction. As we said, in feedback inhibition, an end product of the pathway inhibits one of these steps, what we call upstream in the metabolic pathway. And this is a very important regulatory mechanism in cells. We see this occurring all over the place in cells. And we see it at larger levels too. We see this at the level of the whole organism, where we have inhibitory mechanisms to stop certain processes in the body that we need to turn off. One example is ATP. ATP can inhibit enzymes that are working early on in cellular respiration. We take glucose and we break it down into carbon dioxide and water and generate a lot of ATP from that. If the ATP starts to build up in the cell, we can go back around and turn off that cycle until we use up the ATP that's in the cell. And that way the cell is becoming more efficient. Why burn all that glucose and create ATP, which we already know is an unstable molecule, and it would just go to waste. We would just produce a whole bunch of heat and have no energy gain from that. Now, enzymes are hugely important in our lives. If you think about all the metabolism that occurs is essentially being regulated by enzymes and those factors that influence enzymes. A lot of pharmaceutical drug discovery relies on finding inhibitors to enzymes in specific pathways. If a specific pathway has gotten out of control, one of the ways to go in and control it is to regulate the enzymes that are regulating that pathway. We'll take a look at a video that talks a little bit about this regulation of enzymes to control a particular pathway. This is just one example. There are many, many different examples out there of drugs whose mechanisms work by enzyme regulation. And then we'll see another video that looks at a sort of a whole different aspect of enzymes, and in this case it is in food production.
Within the healthy human body, serotonin is an essential hormone that regulates mood, normal gut motility, cardiac development, and heart rate. However, in patients with metastatic neuroendocrine tumors, functioning net cells act as a factory, overproducing serotonin. Within these cells is tryptophan. Tryptophan binds to tryptophan hydroxylase, called TPH. TPH is the rate-limiting enzyme that converts tryptophan to serotonin, which is then stored in secretory granules inside the cell and released systemically. After being released extracellularly, serotonin is converted to 5-hydroxyindoleacetic acid, called 5-HIAA, a metabolite of serotonin. 5-HIAA is then metabolized in the kidney and excreted as urinary 5-HIAA. The intracellular overproduction of tumor-derived serotonin can eventually flood the extracellular tumor microenvironment, fueling a cascade of symptoms that can include life-altering diarrhea, flushing, bronchospasm, and valvular heart disease, all of which can negatively impact patients' overall health. Historically, the mainstay of neuroendocrine tumor treatment has been somatostatin analogs called SSAs. These agents bind extracellularly to somatostatin receptors 2 and 5 on the neuroendocrine tumor cell, inhibiting the release of serotonin and other vasoconstrictive substances. However, the majority of patients taking SSAs lose their response over time, including 50% within the first year of therapy. Therefore, the overproduction of intracellular serotonin remains unchecked and symptoms may persist, including chronic carcinoid syndrome diarrhea, also called CSD. Serotonin levels are a measure of disease severity in carcinoid syndrome. It's time to take comprehensive control of tumor-derived serotonin by going beyond the surface to target serotonin overproduction where it starts, inside the metastatic net cell. Developed by Lexicon Pharmaceuticals, Zermelo, telotrestat ethyl, is a first-in-class oral TPH inhibitor developed to target and turn down the overproduction of serotonin within neuroendocrine tumor cells. In the metastatic net cells, Zermelo inhibits TPH, thereby decreasing serotonin overproduction. Through the inhibition of TPH, Zermelo reduces the frequency of carcinoid syndrome diarrhea, CSD.
If you've ever made gelatin for a picnic or party, maybe you've been warned not to add certain fresh fruits like kiwi or pineapple. And for good reason. Add these foods to your gelatin and it won't set. It'll remain a big, goopy mess. The reason why is pretty straightforward, but if you understand it, it can help you learn how to make better desserts and how to up your cooking game in general. To understand why these fruits ruin gelatin, you need to know what this stuff is made of. Once it sets, gelatin is basically a mesh of interconnected collagen molecules. Collagen is a long, fibrous protein found in things like skin and bones, and yes, the collagen in gelatin does traditionally come from animals. Manufacturers boil things like hides or bones until the proteins come out, and then they turn them into a powder you can buy at the store for like $1. When you dissolve the powder in hot liquid, the collagen separates into individual fibers. Then as the solution cools, those fibers start to bind to one another and gradually form a mesh. But if you add certain fruits as you're making your gelatin, the last process will never happen. It's because fresh kiwi, pineapple, and some other fruits contain high concentrations of proteases, or proteolytic enzymes. These are substances that digest proteins, basically chopping them into tiny pieces. If this stuff gets into your gelatin mix, it will cut up those strands of collagen until they're too short to link together, so your dessert will come out looking like a soup. If you know why this happens though, it's easy to find some ways to work around it. For one, if you cook the fruit, the heat will inactivate those enzymes, so your gelatin will turn out just fine. This is also why something like canned pineapple doesn't cause issues. Alternatively, you could make your dessert with agar instead of gelatin. Agar is made from certain types of red algae, and like gelatin, it forms an interconnected meshwork of fibers. But it's made of carbohydrates, not proteins, so proteases won't digest it. Of course, while these fruity enzymes aren't great for gelatin, they do have their uses. For one, they have some surprising industrial and medical benefits, like they can be used to prep damaged areas for skin grafts if someone gets a severe burn. The treatment breaks down and clears away dead and damaged tissue. But maybe more importantly to you, You can also use juice or pulp from protease-containing fruits in the kitchen to tenderize tough cuts of meat. The enzymes break down stringy bundles of proteins, making the meat easier to chew. So go ahead and save that fresh pineapple for your steak, and stick to the canned stuff when it comes to gelatin. Everyone will thank you. If you want to learn more science-inspired cooking hacks, we've got an episode with 7 more of them for you. And, as always, thank you for watching this episode of SciShow.
That concludes our section on enzymes and energy transformations in cells.